I'm Kerry Rye. I'm from Sydney, Australia, and the chair of the ATVP Council. And welcome to Science News. And I'm talking today to Marlene Rabinovitch from Stanford University. She has been the distinguished lecturer for the Vascular Discovery meeting this year, and we're going to have chat about the work that she's talked about, the backstory, and where it's heading. So, Marlene, maybe you could start off with telling us a little bit about the sort of overriding of the metabolic phenotypes that you're working on in the pulmonary hypertension space, and the backstory as to how you got to the place you are now.
The thinking initially was really to try to work out, from looking very critically at the structure, this is what I'll talk about tomorrow, the structure of the vessel wall, to see if there was a clue in terms of what was initiating the disease process. And that led to the discovery of an enzyme, and that led to the discovery of an inhibitor. But it took over almost 4 decades until we had an inhibitor that could actually be given safely to patients. So we've, and I'll discuss why we chose this recombinant protein as an inhibitor, and a very serendipitous finding related to the inhibitor, in that it seems to override a genetic mutation that's associated with pulmonary arterial hypertension by amplifying signaling through a deficient receptor. But the storyline behind it is that it has taken a long time from discovery of a process to actually being able to take a compound through a phase 1 and hopefully shortly through a phase 2 clinical trial. And thankfully in the phase 1, there have been no adverse effects. But you never know. No matter how good a treatment appears to be in cultured human cells or in experimental models, the best we have, that it's actually going to work, and in which patients it might be optimal, and at what dose and at what frequency. So with that in mind, while we recognize that we can always improve on these features, we also thought we needed to work on other strategies to develop potential therapies for a disease for which there really, to date, there is no approved modifying therapy of the disease process. There are vasodilators that try to delay the demise of patients and improve symptoms, and have been successful in that regard, but don't really reverse the underlying pathological features that cause the obliteration of lung blood vessels. And that's true in a number of vascular pathologies, not just in pulmonary arterial hypertension. So we approached it in different ways, knowing that the receptor was deficient. Was there a way to directly modify the receptor? And we used high throughput screening to find tacrolimus. But I'll also talk about a strategy that we use to try to look at the abnormality at the cellular level, in agnostic to the mutation, because the receptor is also deficient in patients without the mutation, but less so. So what we really want to do is revert the abnormality at the cellular level. So we did a high throughput drug screen, and actually I'll explain how we found our lead compound, both in terms of its ability to reverse the abnormal phenotype at the cellular level, but also because the compound we found also reversed the abnormal signature that we established bioinformatically in pulmonary hypertension. So it produced an anti-signature. And when we probed why it was so effective relative to other compounds of the same class, we found that indeed it did activate much better the VMPR2 signaling pathway, which is the pathway that's deficient in pulmonary hypertension. So we weren't looking for that, but we ended up finding it. And that helped explain the very beneficial effects of this compound, which could also be repurposed for clinical use, because the compounds we selected were in different stages of clinical development through libraries. And then the third approach we used was really, again, based on a serendipitous finding that transiently, when cells are exposed to low oxygen, BMPR2 is upregulated, except in cells that are deficient. And it's upregulated to send particles or microvesicles into the circulation to support vulnerable cells, to prevent their loss, to prevent their altered change in phenotype, to prevent one of the features in smooth muscle cells is the proliferative phenotype that causes the obliteration of blood vessels. So we figured out why BMPR2 was important in generating these microvesicles, and also we figured out what the cargo was that was BMPR2 dependent, that was so protective. And I'll discuss that in the lecture as well. And how endothelial cells orchestrate the release of these microparticles to support other cell types, support vulnerable endothelial cells, but also to repress abnormal function of smooth muscle cells. So the upside of that is you can take induced pluripotent stem cells, correct the mutation, and then utilize these cells to release microvesicles, and you can actually add these microvesicles back into the circulation to restore normal function of the deficient receptor.
That is really interesting. And so one of the things that struck me just listening to you was whether what you're doing has applications beyond pulmonary hypertension.
In principle, it would. That there are a lot of clues in structure, particularly in the initiation of a disease process that leads you to specific to function to genes, and you can go backwards gene to function to mutations. So I think going back and forth along that pathway and trying to reconcile differences is very important in whatever disease you're studying. And also that there are multiple different ways in which you can, even if you can override the dysfunction that's created by even a very serious mutation, but you can also amplify signaling from a receptor that we know is vulnerable to decreased function in response to multiple different injurious insults. So I think even though the receptor per se hasn't been identified as a gene of interest in systemic vascular disease, although it contributes for sure in experiments that were done by Dr. Zhou at Emory. We can, in fact, look to the strategies that we use to find other compounds that might be beneficial in overriding mutations and variants that are associated with high frequency, let's say, of coronary artery disease or aneurysms or other forms of vascular pathobiologies.
Marlene Rabinovitch, thank you very much for talking to us on Science News. It's been a pleasure having you with us as a distinguished lecturer for Vascular Discovery 2021. And here's hoping that we can have a face-to-face meeting for Vascular Discovery 2022.
I hope so as well. Thanks very much. It's a great honor, so I'm thrilled. Thank you.